Slide 1. Peripheral circulation is a series of slide tape programs developed and produced by the American Physiological Society. This is Local Control Part 1. Content experts, Dr. Brian R. Duling and Dr. Paul C. Johnson. Please note that a printed study guide accompanies this program. It contains the full program text, a test of the concepts presented, and references for additional study. 2. In this program, we introduce the mechanisms contained wholly within tissues and organs that maintain the blood flow necessary to meet the needs of the tissues and enable them to carry out their functions. We discuss in detail the functional and mechanical characteristics of vascular smooth muscle and describe its response to distension. 3. Before we begin this discussion, we should briefly review the topics covered in the earlier programs in this series that are important in understanding the concepts to be presented. In hemodynamics, we discussed the basic principles of fluid flow and examined the factors responsible for dissipation of pressure as expressed in Poiseuille's law. We then applied these principles to an analysis of blood flow, including the factors contributing to both viscous and turbulent resistance and the likelihood of turbulent flow as described by the Reynolds number. 4. In Part 1 of arterial and venous systems, we discussed the organization of the vascular network and the distribution of resistance throughout the network. 5. We also discussed the elastic properties of the vasculature, capacitance, and the force balance between transmural pressure and the circumferential stress described by the Laplace law. 6. In Part 2 of arterial and venous systems, we analyzed arterial systolic and diastolic pressures, the determinants of pulse pressure and mean arterial pressure, the transmission of the arterial pressure pulse wave, and venous blood flow, storage, and return. 7. In Exchange 1, the capillaries and solute diffusion, we described the anatomy of the terminal vessels of the microcirculation and the endothelial histology of capillaries, and we described the process of diffusion of solutes across the capillary wall. 8. In Exchange 2, filtration and absorption of fluids, we described the manner in which fluid moves across capillary walls, the forces that cause fluid movement, and the relationships between these forces and the membrane structure that determine the rate of fluid movement across capillary walls. 9. Your first objective is to be able to describe the functions performed by the peripheral circulation and outline the control systems that regulate blood flow. 10. The peripheral circulation must meet the needs of the individual organs and tissues that it perfuses. In meeting these needs, the peripheral circulation performs a variety of functions. Foremost among these are providing the nutrients required for normal metabolism, especially oxygen and substrates, and removing waste products of metabolism, carbon dioxide, for example. 11. The circulatory system also maintains the normal distribution of fluids within the tissues. 12. Finally, the peripheral circulation transports materials such as foodstuffs from organ to organ and transports hormones from endocrine glands to target organs. 13. Blood flow through the organs of the body is regulated by 3 major types of control systems, all of which act by modifying the contractile behavior of vascular smooth muscle cells. Contractile behavior may be modified by local control mechanisms that originate within the organs themselves or by neural or endocrine systems. The local control systems are the subject of this presentation. 14. Blood flow to individual organs is determined largely by the local control systems that are contained wholly within the organs themselves. Myogenic mechanisms and metabolic mechanisms are responsible for the vascular smooth muscle behavior that results in local control of blood flow. These control mechanisms are sensitive to changes in the internal environment of an organ and respond to maintain the constancy of this environment. 15. Myogenic mechanisms can be seen following a change in arterial pressure. When the pressure rises, for example, the arterioles constrict. This constriction helps to keep flow through the capillaries constant in the face of increased pressure upstream. 16. The metabolic mechanism can be seen following a change in tissue metabolism. When metabolism increases, for example, the tissues release substances that dilate the arterioles and increase blood flow to the organ. The increased flow supplies the increased nutrient needs of the tissues and washes away the waste products produced by the elevated metabolism. 17. What are the principal functions of the peripheral circulation? What are three major types of control systems that regulate blood flow? Please stop the tape while you answer these questions and those that follow throughout the program. 18. The principal functions of the peripheral circulation are to supply nutrients and carry away waste products, to maintain a normal fluid balance in the tissues, to carry hormones from glands to target organs, to transport foodstuffs from organ to organ. The three major control systems are local, neural, and endocrine. 19. What are the two mechanisms responsible for local control? How do they modify flow? 20. Myogenic and metabolic mechanisms are responsible for local control. They couple peripheral circulation to tissue function, which maintains the constancy of the internal environment. The local control systems act by modifying the contractile behavior of vascular smooth muscle cells. 21. Your next objective is to be able to describe the important functional characteristics of vascular smooth muscle cells. 22. Recall that the innermost layer of the arteriolar wall, the intima, is composed of overlapping endothelial cells, and the second layer of the wall, the media, is composed predominantly of smooth muscle cells enmeshed in an elastic network. In an arteriole, there is only one layer of smooth muscle cells, which are 2 to 5 microns wide and from 30 to 100 microns long. 23. Vasoconstriction comes about by active shortening of the contractile machinery in the smooth muscle cells. This shortening process requires energy and is initiated by a stimulus which may be electrical, chemical, or mechanical. 24. Vasodilation results from relaxation of vascular smooth muscle, which may be induced either by an increase in the concentration of a dilator substance or by a decrease in the concentration of a constrictor substance. 25. Perhaps the most important functional characteristic of smooth muscle with regard to regulation of blood flow is that it can contract spontaneously and can sustain submaximal contraction for long periods of time. This contraction is referred to as vascular smooth muscle tone. It is important because it provides a background against which vasoactive substances can operate. 26. Vascular smooth muscle tone arises from a variety of sources. It can be induced, for example, by a low-level discharge of sympathetic neurons, which release norepinephrine. This is referred to as neural tone. 27. Vascular smooth muscle tone may reflect the net result of a variety of inputs. That is, it can sum the actions of a number of stimulants and relaxants, assuming an intermediate state of contraction. 28. Smooth muscle cells can be grouped broadly into two types, mainly on the basis of their membrane characteristics. Single-unit smooth muscle possesses good electrical continuity between cells and generates spontaneous action potentials. Therefore, it contracts as a unit and produces a high level of spontaneous contraction. 29. Multi-unit smooth muscle cells, as the name implies, tend to function independently of each other, essentially as individual contractile elements. Multi-unit cells do not usually produce action potential spikes and do not propagate action potentials between adjacent cells. In terms of regulation, the most important characteristic of multi-unit tissues is that the cells produce few spontaneous contractions. We should emphasize that these are broad categories. The distribution of vascular smooth muscle types is not uniform, and cells from various regions of the circulation show a wide range of behavior. 30. What is meant by the term smooth muscle tone? What type of smooth muscle produces both action potentials and spontaneous tone? Name two types of changes that cause vascular smooth muscle relaxation under physiological conditions. 31. Smooth muscle tone is a sustained submaximal contraction. Single-unit smooth muscle produces action potentials and spontaneous tone. Vascular smooth muscle relaxation is caused by an increase in vasodilator substances or a decrease in vasoconstrictor substances. 32. Your next objective is to be able to discuss the role of calcium in determining vascular smooth muscle contractile strength. 33. To a large extent, the contractile strength of vascular smooth muscle is determined by intracellular calcium ion concentrations. Most agents that modify contractile strength act by modifying the intracellular concentration of calcium ions. Single-unit and multi-unit cells differ, however, in the sources from which they derive the calcium involved in contraction. 34. Single-unit cells tend to depend on an influx of extracellular calcium. Stretch, for example, seems to induce contraction by increasing transmembrane calcium influx. This contraction, called the myogenic response, is discussed in the next section. 35. Multi-unit cells depend mainly on release of intracellular calcium, stored in sites such as inner sarcolemmal membranes and endoplasmic reticulum. 36. Vasoactive agents such as norepinephrine can modulate intracellular calcium by acting at a variety of sites. They may modulate transmembrane flux patterns, or they may modulate intracellular release from endoplasmic reticulum or inner membrane surfaces. 37. What is the ultimate determinant of contractile strength in smooth muscle? What is the primary source from which the calcium involved in contraction is derived in single-unit cells, in multi-unit cells? 38. The ultimate determinant of the contractile strength of smooth muscle is the intracellular concentration of calcium ions. Single-unit cells depend primarily on extracellular calcium. Multi-unit cells depend mainly on release of intracellular calcium stores. 39. Your last objective is to be able to describe the vascular smooth muscle response to distension. 40. The special mechanical characteristics of the smooth muscle determine to a great extent how the vessels are influenced by pressure and changes in pressure. By mechanical characteristics, we mean the relation between stretching forces applied to the vascular smooth muscle cell and the degree of deformation induced. 41. Recall that the stress in the vascular wall is equal to the product of intravascular pressure and the ratio of the vessel's radius to its wall thickness. Regardless of whether the vascular smooth muscle is the single unit or multi-unit type, it must continually resist distension caused by the pressure within the vessel. 42. When intravascular pressure increases, the vessel dilates, the smooth muscle stretches, much as a rubber band, and the stress in the wall increases. This elastic property is the simplest mechanical characteristic of vascular smooth muscle. 43. The second important mechanical property of vascular smooth muscle is its viscous behavior. When pulled upon, it stretches initially, then undergoes a slow elongation. The phase of slow elongation is called creep. 44. Since vascular smooth muscle shows both viscous and elastic behavior, it is called a viscoelastic material. To reiterate graphically, if the pressure inside a vessel suddenly rises, there is an immediate increase in the radius of the vessel. This reflects stretch of the elastic elements in the wall. Stretch is followed by a much slower distension, mainly as a result of slow lengthening or creep of the smooth muscle contractile system. 45. In some vascular smooth muscle, the stretch caused by increased intravascular pressure triggers the myogenic response, which leads to sustained contraction of the smooth muscle cells for as long as the pressure remains elevated. 46. Creep and the myogenic response tend to induce opposite length changes in smooth muscle cells. Creep results in a slow elongation, and the myogenic response results in a relatively slow shortening. 47. Since the myogenic response tends to offset creep, the net effect of increasing the intraluminal pressure in blood vessels may be quite variable, depending upon the sum of the two processes. 48. The myogenic response is much less active in multi-unit muscle than in single-unit muscle. Therefore, in large vessels such as the carotid artery, which possesses primarily multi-unit muscle, an increase in distending pressure tends to induce creep. 49. In contrast, in small arterioles that possess single-unit smooth muscle, an increase in distending pressure is more likely to induce the myogenic response. In some vessels, this response may be so marked that the radius of the vessel actually falls below its initial size. This is important in the myogenic mechanism for control of blood flow. 50. Show algebraically how pressure inside a blood vessel can be related to distension and stress in the wall of the vessel. 51. Stress in the wall is determined by the pressure, vessel size, and geometry. Specifically, stress equals pressure times radius over wall thickness. 52. Differentiate graphically between elastic deformation and creep. 53. Elastic deformation is the immediate change in size of a vessel induced by stretching the wall elastic elements as intraluminal pressure is raised. Creep is a slow elongation which results from slippage of partially activated smooth muscle contractile elements. 54. On your graph, show how the myogenic response acts to stabilize vessel radius when luminal pressure is raised. 55. The myogenic response induces contraction or shortening of smooth muscle cells when the tissue is stretched, thereby generating a force that counteracts the deformation induced by the increased pressure. 56. This concludes part 1 of local control. We discussed the functions of the peripheral circulation, described the local mechanisms that couple peripheral circulation to tissue function, discussed the role of calcium in determining smooth muscle contractile strength, and described the details of the myogenic response. It is important to remember that the myogenic response is a regulatory process residing in the nature of smooth muscle cells, especially in their active response to increased circumferential tension or stress. To review these concepts and for a list of additional readings, please refer to your study guide. 57. In Local Control Part 2, we discuss the metabolic interactions between the tissue and vasculature that balance blood flow and tissue function, and we discuss in detail the combined roles of myogenic and metabolic mechanisms in three major phenomena: functional hyperemia, reactive hyperemia, and autoregulation. 58. This program is part of the Peripheral Circulation Series. It was developed and produced by the American Physiological Society. Copyright 1981.